most important tools used in a biology course is a microscope. Today we'll be talking about some tips for purchasing microscopes and also 5 easy steps for focusing a compound light microscope like this one. Come join us! There are 2 major types of microscopes commonly used in a middle school or high school science classroom. The first one is a stereo microscope or dissecting scope, and the second is a compound light microscope. We'll start with the stereoscope. The stereo microscope is a great choice for students just entering middle school, or if you have younger elementary school age children at home that may also be using it. It's quick to use and will generate scientific interest with your children extremely quickly. They can place any 3-dimensional object under the scope and be able to focus it in just seconds. Unfortunately, the downside of the stereo microscope is that the magnification level is only up to about 50 times. This may seem like plenty of magnification, but for upper level science courses like high school biology and some middle school life science courses, they'll need to be able to focus and see on a cellular level, which will require a compound light microscope like this one. Another note if you choose to purchase a stereo or dissecting microscope. If you have young children, because of the 2 eyepieces, they may not actually be able to see into both eyepieces at the same time, based on the distance between their eyes. If this is the case, I suggest that they just use the 1 eyepiece until they grow into the microscope. Light microscopes are perfect for looking at organisms on the cellular level, such as pond organisms, bacteria, or human cells. Light microscopes have a wide range of magnification levels, and some boast up to 2,500 times magnification, which is almost never necessary for a high school biology course. The total magnification of a light microscope is determined by 2 lenses, one in the eyepiece and one in the objective lens. The total magnification of a light microscope is calculated by taking the magnification of the eyepiece times the magnification of the objective lens. So for this typical microscope, the eyepiece has 10 times magnification and the objective lens has 40 times magnification, giving us a total magnification of 400 times. Most typical light microscopes for high school courses have 3 objective lenses, ranging from 4 times to 100 times magnification, giving the total light microscope magnification somewhere around 1,000 times. The other thing to consider when purchasing a light microscope is that the light source is below the stage and projected upward. So unlike a stereo or dissecting microscope, which can look at 3-dimensional objects, a compound light microscope requires a thin specimen that the light can pass through. This is why pre-made slides from biology companies are so popular. But I recommend that you don't shy away from making your own slides, as it's not as difficult as it seems. The other difficulty that many students have with compound light microscopes is focusing on the slides, as it's significantly more difficult than with a stereo microscope. Thankfully, in the second portion of this video, we'll be looking at 5 easy steps for focusing on a slide. There are 2 knobs that are used to focus on a specimen using a compound light microscope. The first is the coarse adjustment knob, or coarse focus, and the second is the fine focus, or fine adjustment knob. Your knobs may be located higher or lower on your microscope based on the manufacturer, but the coarse adjustment knob should always be larger and the fine adjustment knob is always smaller. The purpose of these knobs is to move the stage up and down, bringing the specimen into focus. The coarse adjustment knob is always used first and only on the lower 2 powered lenses. The fine focus knob is used second and brings the specimen into finer detail, and is the only knob that should be used on your highest power objective lens. So, let's focus on a slide. Okay, step 1 is to move to your lowest power objective, which would be your shortest. and move your stage with the coarse adjustment knob all the way to the bottom. Step number 2 is to put your slide on the stage. I have a handy stage clip here. If you don't have one of those, that's fine. You just need to be careful not to bump your slide and move it while you're adjusting your focus. Step number 3 is to position your specimen so that it's directly over the hole where the light is passing through. Step number 4 is to look into the microscope and adjust with the coarse focus knob, which is your larger one, until the specimen becomes clear. Don't go too quickly or you'll pass the point of clarity and then you'll be stuck again. Okay, I found it. Now I use one eye open, one eye closed when I focus my microscope. Technically that's not the way you're supposed to do it. You're supposed to keep both eyes open, but it's been working for me so far, so I'm going to keep doing it. Step number 5 is simply to use your fine focus knob to bring your specimen into the greatest detail possible. You shouldn't have to adjust this very much. Also make a note that if you're seeing your eyelashes in the eyepiece, that means you need to back up. So now your specimen should be as focused and clear as possible on low power. However, it may not be as magnified as you need. So without adjusting the coarse and fine focus again, move to your medium power objective. Now you're going to want to look through your eyepiece and use your coarse and fine focus again to bring it into greater clarity. If you can't see your specimen anymore now that you're on medium power, you need to go back to your low powered lens and center it a little bit more. Because with each greater magnification and each new objective lens, the field of view gets smaller and smaller. So if your specimen is not directly in the middle of the hole where the light is passing through, you won't be able to see it as you move to your different objective lenses. Also, note, very importantly, that when you get up to your high-powered objective lens, as I mentioned before, you only want to be using your fine focus knob. At that point, the specimen should be pretty clear anyway, but if you use your coarse adjustment knob, you can actually push your stage and slide into the lens and it'll scratch your high-powered objective lens, which is absolutely not what you want to do. So I hope this has been helpful, and I hope that the next time that you need to focus on a slide with a light microscope, it'll be a lot easier.